Greetings everyone. We're here today with Dr. Fabien and Nikhil. Nikhil is a new dad of a Fox G1 Bob in the States, and he is an incredible programmer who's helping us with our using the TERRA platform, and he has understood it extremely well to the point he was able to explain everything to Fabien, who is going to use this in reality. So, Fabien, do you want to introduce yourself briefly, and then we'll talk about what we've been up to?
Sure. Hi everyone. So, my name is Fabien. I am the director of the transgenic facility here at Macquarie University in Sydney, and part of my job is to build models for other researchers to find what's going on when diseases kicks in, but also to find obviously cures and new drugs to treat these disorders. And a lot of what I do relates to genetics, where we kind of tweak the DNA. And one of the main problems that we have is that the technologies that we use to read the DNA, the genome, are called sequencer, and this technology keeps evolving. And lately, one such technology is called long-read sequencing. This technology came out not so long ago, and we acquired such technology and developed a workflow. But the problem we have is how to interpret the data. The data that are generated is something that really needs to be analyzed. And for that, we need someone like Nikhil, who is an expert in programming, to be able to put all the analysis package, the software that are needed to work on this data, to actually get an output, a simple output that speaks to us and that we can translate in terms of whether, for instance, an experiment worked or not. So luckily, you, Steve, put me in touch with Nikhil, and Nikhil applied his magic recently, and to say the least, I've been blown away, because this is something that requires a lot of expertise. And I thought, I shouldn't say that, but I thought no one could achieve, and particularly in such a short time frame. So maybe, Nikhil, we can talk about what exactly the question was and what you did. Maybe let me start with what exactly the question was, because I think it's going to be relevant to other researchers. So I had run an experiment with long-read sequencing, and my problem was the raw data I was absolutely unable to analyze. But what I knew is this long-read sequencing was used to kind of check whether I inserted a piece of DNA in the genome of some cells, and I called you and I said, or actually I called Steve, and Steve called you and said, can you make that happen? And then from there, you took over, and you may want to explain really simply what you did. I believe you have a diagram that simply explains the workflow of what you built, knowing that the idea is for people like me to actually not have any knowledge of what's going on in the background in terms of the programming, but just to have an output that is understandable to us.
Sure, Fabien, yeah. That's a good introduction, basically. So I think the problem with processing the long-read sequencing data is it's kind of very new, right? And there are not a lot of standardized workflows or pipelines which are available to run that. So, and I think that's why I think when we started looking into TERRA, what we found out is that there are already few pipelines developed by folks at Broad Institute for ONT, as well as the PacBio sequencing data which comes out, all we had to do is basically made some changes to it or tweaks to it to include the steps you wanted to run on that data, right? So what I can do, I can share the screen and I can show you the two major steps that are required for running this, the processing on the ONT sequencing data. One is the base calling step using Guppy, and the second is the de novo assembly using Canoe. So let me share my screen and I can quickly show the diagram to show all the steps that are involved.
Just Nikhil, just quickly, I'll just jump in very, very quickly. Fabien, what you were trying to do was verify a knock-in. Was that correct?
That's right. So what I call inserting of piece of DNA in a targeted fashion. So at a very location that I know in advance, I target it. Is called knocking in this piece of DNA. So I was trying to check whether this knocking was actually here or not.
Yeah, so this is a great verification step to know whether that is a viable model or not.
If it happened in the first place,
yeah.
If it happened properly, and lately if there's something else that happened or not, that may or may not be what I expected.
Yep, cool.
Correct. So I think the two steps, right? One is the ONT base call and the second is the ONT assemble. So if you open the ONT base call step, and I think in the last demo we went through how to run it and what are the input. So we can look at probably what are the steps involved to run this, right? So yeah, if you see here, we start with the input FAST5 files, right, which are coming out of the sequencing, and these are the various steps that are basically involved, right? And the great thing about TERRA is that once you configure those steps, this diagram is generated automatically and updated also every time you make some changes. So just provides kind of a nice visualization, right? Whoever is actually running the workflow directly without knowing underlying mechanics, as Steve says, like underlying what's going on, but just knowing that these are the steps I want to execute and those steps are getting executed or not.
Yes. So what I found really amazing, and I think you might also want to share in the end this graph that you just showed me before. But just before you do that, the idea is I give you the raw data, which in this case are the FAST5 files, and this is really the only input that a researcher like me needs to have, an input into TERRA, and then your workflow will run and eventually gives even a graphical output, which to me makes sense, and maybe you can share it now. And that, in a second, I can look at it, and I can already know whether this knocking is here, if, for instance, or it's not, to what extent it is here or not, in terms of proportion, and also if there are some other mutations, for instance, that have happened around this insertion. So being able to only provide FASTQ files, which anyone, even I, can do that. I can copy that and put it into the workflow in TERRA, and getting in a very short time frame without having to do anything but pressing a button, being able to have an output which is a graphical output is really something for me that's life-changing in terms of enabling the research to happen. Because to get something like that, usually researchers need to rely on a team of bioinformaticians that they would collaborate with or find a way to interact with, and this team would need to set up the workflow and work on each time on the input with the new data set, and at the end of the day, there's no knowledge transfer, because I would only always have to rely on a team of bioinformaticians, whereas here you made it available to pretty much everyone. And anyone with barely any knowledge of genetics can actually get a result, which is absolutely mind blowing.
The other thing that's worth saying here, Fabien, is if a new tool became available, you would just stick it in here, right? It's not like you're having to learn how to program and download dependencies and have all these, Windows open. You just, like someone like Nikhil could very easily bring that tool in, and this actually is a perfect example where it's called GPIDE. This application is called GPIDE, and I remember when you and I were talking initially, you had it on your laptop, and so you were showing us this output in that software, and then Nikhil and I spoke about it, and I said, well, can we put GPIDE in the workflow so that you don't even need that software anymore? And he just did it. He just figured it out and did it.
Exactly, and so now you see this graph at the bottom is something that tells me immediately whether my experiment failed or succeeded. This is something incredible, because without that, I am unable to tell whether it worked or not. And as you mentioned, Steve, because there is this flexibility in changing the workflow, it is relevant to my very specific experiment that we visualize here, which is whether the knock-in is here or not. But it's going to be relevant to anyone using this long-read sequencing technology. And obviously, there are people addressing different research questions, which, for instance, can be trying to identify new mutations in patients who have no diagnostic because we haven't found yet in their genome what's going on. This sort of workflow will enable geneticists around the world to have the answer to their research question, which is in a very simple and easy way. So it's kind of incredible, really, that Nikhil managed with barely any knowledge of the genetics behind. Yeah. I have that knowledge, but I have no knowledge of the programming, and coming together, having this output is just incredible.
Yeah, and that was always in my mind when I initially looked at TERRA, when I understood from FRF globally that we had all this data, and then I started learning about how the analysis is done, and it's all in many CSV files, and these people have this little bit of data, and these people have this little bit of data, and no one sort of has a collection of all the data. And then it's all these hand-bashed tools, and I remember thinking, like, why should a genetic or any sort of medical researcher have to be a software programmer? And this just fixes that. Like, that should be pulled away from each other, and that is exactly what this does. It does all the work that you don't need to do. I love it. I think it's amazing. It's incredible.
The reality is that we're not programmer, and even if we try
and you shouldn't be expected to be.
Exactly, but I still try because I have such a need, and it's a growing need. We need more and more this expertise. But I failed because we're so already busy on focusing on the research question. Actually, being at the same time having the expertise that Nikhil has, let's put it like that. It's impossible. It's incredible what Nikhil has been able to do, and putting it in TERRA, all integrated in the one place like that. It's incredible.
Great.
Right, and this was the case with the long-read sequencing, Fabien, because this was fairly new. So we had to make kind of some changes or some tweaks to the workflows to match what you needed. But with respect to the NGS sequencing analysis, right, there are already so many workflows that are available. So if I go to the library of the featured workspaces or the workflow, so this takes me to the doc store, and this is kind of a collection of workflows. So if we see the amount of workflows which are written for NGS or long read sequencing, which are readily available for researchers to import into TERRA and run the analysis with their data, is huge, right? So we don't have, in order to make any changes to the workflow, basically just import it into your workspace and run it with your data and analyze.
Yeah, that is because the TERRA has a second advantage that you just showed, is that NGS is a technology that's a bit older than long-read sequencing. Therefore, some people have worked out some part of the analysis. But what's amazing is that everything is already all put together in TERRA, and then it's up to you and me to discuss and see how to organize the workflow.
And it's free.
And it's free.
Yeah. And it can be shared. So if someone had a very similar use case and had a small problem, they have a good community where you could say, I have this little problem, and someone with Nikhil's kind of skills can, in many cases, just fix it.
Absolutely.
Yep. Yeah, and we published, the work that we did with our workflows, and that should also be available here, basically. So if we see, even the workflows that we created, those are available as, the published workflows, basically, in DocStore. So if somebody wants to run very similar workflow, they don't have to make those changes. They can just, import that, right? View public page. They can just click on this TERRA button, and it lets you import into your work. So you choose the workspace you want, and then basically it just imported, and then it's available basically under the workflow session for you to run. Yeah.
And I think it's just important to say that's without our data. That's just the workflow, just in case anyone was worried about that. They're only going to see the workflow, not the data.
Mm-hmm. Yeah.
Great. All right. Listen, let's not make this a really long one. Did anyone have any final things they wanted to say?
Yes, I want to say thank you to you, Steve, and to you, Nikhil. My hats off to you, because having been able to actually set up all this in clearly no time, you did that in few days. It's really helping people like me in their daily work. So thank you very much, and I'm sure we'll keep in touch, and as the technology evolves, I'll knock at your door more than once for sure in the future, and I suspect I won't be the only one once this video is published.
I hope so. I also want to say thank you, Nikhil, very much. That is just unbelievable. I cannot believe that someone with no background in this has just answered the questions that Fabien needed. It's perfect. Well done. Really well done.
Absolutely.
Right, and that kind of speaks about usability of this platform also.
Yeah.
So I'm coming from a different domain. So yes, I'm software programmer, software engineer, but genetics domain is new to me, right? So it was very easy for me to transfer my the technical knowledge into this, right? So that speaks about how usable and user friendly this platform is, basically, for researchers as well as people who want to use it.
Well, let's share this video far and wide and make sure that everyone that can benefit from it gets it in front of their eyes. And I suppose if anyone is interested in looking further, you can Google TERRA. It's built by the Broad Institute, and I hesitate to say, but contact me if you need, and I'll try and put you in touch with Nikhil or someone that may be able to help you. But I feel like my inbox might get filled up pretty quickly. So we'll see how that goes. But in any event, thank you again, fellas. Yeah, great job.